Hello everybody, my name is Hassan Nafid. I am an anatomy and physiology instructor at Madison College. I wanted to welcome you back again to our anatomy sessions, and this time we're going to talk about one of the activities of our body cells. This activity helps our body cells and body tissues to grow. It helps our body to repair. It also helps our body to regenerate and compensate for lost or damaged tissues. We're going to talk about cell division. We identified two types of cell division: mitosis and meiosis. Mitosis is a type of cell division that takes place in almost every cell in our body, while meiosis, on the other hand, takes place in the gonads or the reproductive organs. In particular, it takes place in the testes and the ovaries. Mitosis is a type of division in which the chromosomes are doubled, while in meiosis the chromosomes are reduced or cut into halves. Mitosis produces cells that are similar to the mother cell, with the same number of chromosomes and almost performing the same function, while meiosis produces a type of cells that has half the number of chromosomes, and these are the cells that are involved in the reproduction. It produces sperms and eggs. We are going to talk tonight and describe the phases of mitosis. In a normal situation, our body cells are either in a state of normal metabolic activity and normal metabolic status, or they are entering or engaged in mitosis. I have some models here that I'm going to use to show you and walk you through the steps of mitosis. I'm going to pick one of the models here, share with you. Position of the camera here a little bit. If you take a look at the cell, I want to also emphasize on something. This activity of cell division takes place in the nucleus. So the nucleus is the center of activity of meiosis or mitosis. The first stage here is known as the interphase. This is the cell stage of metabolic activity and growth, but the cell is not in an actual status of division. If you take a look at the nucleus, the nuclear membrane is intact, so the nuclear envelope is present, and the chromosomes are coiled, they're long, and they are thread-like strands of DNAs and proteins. Chromosomes are not visible in this stage, except as chromatin granules. Under the microscope, the nucleus is going to stain dark purple, while the cytoplasm around it is going to stain light pink. The cell at this stage is not dividing, as we said, but might get ready to divide and move to the second stage. So the distinct features of this stage is the presence of the nuclear envelope and the presence of ill-defined chromosomes. The next stage is going to be the prophase. And in the stage of the prophase, the features are clear visible chromosomes, the absence of the nuclear envelope. The nuclei or the nucleolus will disappear, and the chromatids are present. So each of the chromosomes will split into two chromatids that are held together by the centromere. The chromatids represent the two DNA molecules that makes up the chromosome. Chromosomes will start to move towards the equator of the nucleus. This stage is known as the prophase. The third stage that we are going to enter is the stage where the chromosomes or the chromatids are in the center. As you see, the centrioles will migrate and the spindles formation is complete. Look at the spindles here, radiating the two sides, holding to the chromatids, which are held in the center by the centromere. And here are the centrioles that migrated to the two poles. Note the alignment of the chromatids or the chromosomes in the center. This stage is known as the metaphase. Next step that the cell or the mitosis is going to move to is going to be the anaphase. Take a look at the anaphase here. The chromosomes, as you see, will split apart at the centromere and they're moving away from each other. Look at the chromosomes that are splitting apart, moving to each pole, separating the two chromatids. The separate chromatids are now considered to be daughter chromosomes. These are the future chromosomes of the future two daughter cells. Daughter chromosomes move gradually towards the opposite side or the opposite pole of the spindle. By late anaphase, the two groups of daughter chromosomes are completely moved to the opposite poles of the spindle, and by the late anaphase, cytokinesis has often begun. As you see here, the cytoplasm will start to narrow or split or deepen in the middle, announcing the beginning of the division of the two future two daughter cells. The next stage that the cell is going to move to is the telophase. And if you take a look at the telophase, the cytokinesis becomes more obvious and continues gradually until it is complete, separating the cell into two smaller daughter cells. The chromosomes appear gradually and become less distinct as they are on their way to assume the mother organization of the nucleus. They are uncoiled back into chromatin. The spindle fibers disappear. As you see, you don't see any spindles here. The nucleolus will appear or reappear, and the nuclear membrane will be formed again, which is obvious in this model here. I'm going to share with you. If you look at this model here, the two daughter cells are almost established. Each one has a nucleus with a nuclear membrane, nucleolus in the middle, chromosomes and coiled, indistinct, no spindles, and the cells are almost dividing into two daughter cells. And this will mark the end of the mitosis. It is important to say cell divide at different rates depending on the need for that division, the site of the cell activity. Different cells divide at different rates. For example, red blood cells are very fast dividing cells at a rate of 2 million red blood cells per second. This is a very fast rate simply because there is a high need for larger amounts or larger number of red blood cells to carry oxygen and to transport gases, while some cells are slow growing or are poorly growing due to many other factors. Nervous systems, nerve tissues, cardiac tissues, these are poorly growing or actually not growing or not dividing cells. One of the facts is due to the absence of the centrioles, which are the two organelles that lead the mitotic activity inside the cells. Thank you for watching this video, and I'll meet you in another video of our anatomy sessions.